Hello friends, welcome to my channel Clinical Biochemistry with Dr. P.K. Prabhakar. Today I am going to talk about this is second lecture on the enzyme. So where we are going to see the details about the classification of enzymes. Uh, Actually in this lecture we will talk about the detail about all the classes and their subclasses. However, only the first portion is applicable for the UG and PG students. But if you want to understand the detailed classes of all the enzymes, uh, you should see all the complete video till the end. So you will understand what are the enzymes, what are their class, subclass, and what is their applications. So let's start. Uh, Before going further, if you have not subscribed my channel, you can subscribe it. If you first time, you can click the bell button so that all the notification will come to you. Uh, so today lecture we are going to see all the 7 classes of enzymes, where we will see the oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase, and translocase. And then we will see all these 7 enzymes are going to be have their nomenclature. So before going to see the classifications, we will see the basics of the enzymes. So as we have seen in my previous video, where we have discussed about the enzymes and their introductions. Enzymes are normally it's a biocatalyst, which is also bio means life and catalysis means the anything which normally increases the rate of reaction. So that we are going to call the catalyst of life. If you see the basic definition of enzymes, so enzymes may be defined as biocatalysts, which normally synthesize in the living cells. And chemically, they are protein in nature. Almost all the enzymes are protein in nature, except RNA, which works as an enzyme that we are going to call as ribozymes, which is normally used during the RNA processing, especially transcription processes. So that is ribozymes, except that one, all enzymes are protein. Then we will say the normal. If you see the nomenclature of enzymes, normally we didn't have initially we didn't have any actual system for the nomenclature. So the initially all the enzymes were done the nomenclature normally randomly arbitrary nomenclature had been given like pepsin, trypsin, chymotrypsins. So there are no system for that one. Normally, if you see the enzymes name, if we are going to have the ASE in the last as a suffix on the names, we consider them as enzymes. And sometimes we had a nomenclature like we had a substrate name. We are going to add ASE. That's lactose. We will add ASE, so it becomes lactase. So similarly, galactase, like that one, maltase. So those were the called as trivial nomenclature systems. Then in IUB, that was the you can call it the international system. International Union of Biochemistry. They have appointed an enzyme commission in 1961. Sorry, and this IUB system of enzyme classification came into the picture in 1964. At that moment of time, IUB system of enzyme classification had only 6 classes. So that was oxidoreductase, O, then T, transferase, H, hydrolase, then lyase, L, isomerase, then ligase. So these were the 6 classes, and later on we had a 7th class that is called as translocase. So currently we are having the total 7 classes, but initially when in 1964 we had only 6 classes of enzymes. These enzymes were we have seen in the previous video also that enzymes had normally going to classify on the basis of their location. So we had intracellular enzymes and extracellular enzymes. Intracellular normally they synthesize and their functions are normally inside the cell, inside the plasma membrane. Where they are synthesized, they are normally their function will be at that location only. They are called as intracellular enzymes. And extracellular enzymes are normally all the enzymes, almost all the enzymes are synthesized inside the cells, inside the plasma membrane. But if their functions physiologically, they for their physiological role, if they come out of the cells and their functions will be outside the plasma membrane, outside the cells, they are called as extracellular enzymes. Almost all the digestive enzymes are extracellular enzymes because their function will be normally in our GIT tract. So this was one of the system of nomenclature. Then we had total 7 classes. So these were the 7 classes where we have seen first class was, and this is sequentially you need to remember it because The first class is oxidoreductase, and when we are going to do the IUPAC nomenclature of the enzymes, so it starts with 1 point. Then we are having a transferase, enzyme, that is class 2. Then class 3 is hydrolase, class 4 is lyase, class 5 is isomerase, class 6 is ligases, and class 7 is translocases. And this is the example, this is the normal which type of chemical reaction they performs. This up to this one we have already seen in my previous video, where we have talked about the basics of the enzymes. So class 1 enzymes, which is oxidoreductase, normally perform the redox reaction, that is oxidation reduction reactions, and it can be categorized as oxidase and reductase, where when two molecules is going to react together, one molecule will be oxidized, another will be reduced. So this such type of reaction where oxidation reduction reaction occurs, that is called as oxidoreductase reaction, and enzymes are called as oxidase or reductase. Then we are having transferase, where catalyzes the transfer or exchange of certain groups among some substrates. Like here, if you see, the AB is a one molecule which is going to react with the C. So one portion of this molecule will be AB will be transferred to C, and so it will be B, C, and A will be separated out. So this is called as B is transferred from here to here. So this is called as transferase. Then hydrolase, where hydrolysis of the substance will be there. Normally digestive reactions are hydrolases reactions. Then we are having lyase, where removal of certain groups will be there. Either ammonia you are going to remove, water you are going to remove, carbon dioxide you are going to remove. So some molecules will be removed without the involvement of water. So that is called as lyase reactions. And then we are having isomerase, where normally facilitate the conversion of isomers. Either it will be geometrical or it will be optical isomers. So their structural orientation, three-dimensional structure is going to be changed. Ligases are when they are going to involve in the joining of two or more than two molecules, and translocases are for the transferring of certain molecules across the membrane. So that is called as translocases. So this seventh class has been added later on. Initially we had only these 6 classes. So now we are having total 7 classes of the enzymes. Now one by one we will see. The first class, if we will see, oxidoreductase. The oxidoreductase consists of a large class of enzymes which normally are going to catalyze the transfer of electron from an electron donor. So it is a very large class, which because maximum reaction we are having, that is oxidation reduction reacts. So here we are going to be a transfer of electron from an electron donor, which is normally going to call as reductant, and that electron is going to be accepted by a molecule that is called as oxidant molecule. So we will have a reductant, and we are going to have oxidant reactions. And in these reactions, normally we require certain components, certain factors, certain you can call it the cofactors, and such those are nicotinamide adenine dinucleotide NADH, NADP or NAD plus as a cofactor. Since so many chemical and biochemical transformation comprises oxidation reduction process, it has a long been important in the goal in the biotechnology to develop a practical biocatalytic application of oxidoreductase molecules. During the past few years, significant breakthrough has been made in the development of oxidation-based diagnostic tools or diagnostic tests and improved biosensor, and the design of innovative system for the regeneration of essential coenzymes. So research is going on. Research on the construction of bioreactor or pollutant degradations or biomass processing is going on. Normally, this type of enzymes have nomenclature are in the form of donor, which donates electron. Then colon, we are having accepted name, and oxidoreductase we are going to add it, or it may in most cases donor dehydrogenase we are going to call it. Sometime we are going to have acceptor reductase. So these are the 3 different ways normally how we are going to put the name of this type of enzymes, either donor acceptor oxidoreductase or donor dehydrogenase or acceptor reductase. So in this case, such as NAD plus reductase. Donor oxidase is a special case where oxygen serves as the acceptor molecule. Oxidoreductase can be all oxidase or dehydrogenases. Oxidases are the enzymes which normally involve when molecular oxygen accepts the electron as an or acceptor of hydrogen or electron. So, and dehydrogenase are those enzymes which oxidizes a substance by transferring hydrogen to an acceptor, that is either NAD plus or NAD plus or a flavin enzymes. Normally, if you see the chemical reactions where oxidation reaction takes place, there are four different ways by which oxidation reaction occurs. Either you can define oxidation or reactions on the basis of oxygen transfer, or hydrogen transfer, or electron transfer, or oxidation number transfer. So oxidases are enzymes which normally involve when molecular oxygen acts as acceptor of hydrogen or electron. So the here oxygen is the acceptor molecules. Such type of enzymes where oxygen will work as acceptor, they are called as oxidases enzyme. And dehydrogenase are enzymes where the transferring hydrogen to the acceptor molecule, and the acceptor molecule will be either NAD or NAD plus. So if oxygen is the acceptor, it is oxidases. If NAD plus or NADP plus or flavin is the acceptor, it is called as dehydrogenases enzyme. Other oxidoreductases also named like peroxidases, hydroxylases, or oxygenases or reductases. These are some of the other oxidoreductase enzymes. Peroxidases, which is normally present in the peroxisomes and catalyze the reduction of hydrogen peroxide. Hydroxylases add hydroxyl group into the substance. Oxygenases incorporate oxygen from molecular oxygen into the organic substances. And reductases catalyze the reduction of, in most cases of reductases, acts like an oxidase. So these are the different types of oxidoreductase enzymes. Common category are oxidases and dehydrogenases, but there are some other category like peroxidase, which works on hydrogen peroxide. Hydroxylases, normally hydroxyl group addition to the substance. Oxygenases normally incorporate oxygen molecules, and reductases called reductions. These are some of the common classification of subclasses of oxidoreductase. So the first subclass is 1.1, which acts on the CHOH group or donor. Second one is 1.2, that acts on the aldehyde or oxo group of donor. 1.3 functions as a CH group or donor. So depending on the different types of oxidation reduction reactions, we are having different subcategories of oxidoreductase reactions. Normally, this is the chemical, one of the example of chemical reaction where two molecules react together, one will be oxidized and another will be reduced. So the catalyzed reactions are similar in the following reaction in above figures, where A is the reductant and B is the oxidant. In the biochemical reaction, the redox reactions are sometimes more difficult to observe, such as the reaction from the glycolysis. Here, if you see the glyceraldehyde, where NAD plus is going to work as a cofactor. So this is going to give means of hydrogen, it will be coming from here. So hydrogen will be accepted by NAD. So it becomes NAD plus and NAD. So this is the reduced product, and this glycerol layer 3 phosphate is going to be oxidized. So here NAD plus is the oxidant, that is electron acceptor, and glycerol 3 phosphate works as a reductant, electron donor. And this here the enzyme which works as, that is because this is NAD is going to involve as an electron acceptor, so enzyme will be dehydrogenase enzyme. Some of the common functions of this type of enzymes are, these enzymes normally play a significant role in aerobic and anaerobic metabolic processes. They can be found in biological processes like glycolysis, TCA cycle, oxidative phosphorylation, and amino acid metabolic processes. In glycolysis, the glycerol 3-phosphate dehydrogenase, which normally accelerates the reduction of NAD plus to NADH. Just now we have seen the glycerol 3-phosphate dehydrogenase reaction in the previous slides. So that was catalyzed by glycerol 3-phosphate dehydrogenase. The reoxidation of the NAD plus to NAD plus occurs in the oxidative phosphorylation pathway. Additional NADH molecules are produced during the TCA cycle. There also we are having dehydrogenase enzymes. The glycolytic product pyruvate takes part in the TCA cycle in the form acetyl coenzyme. During anaerobic glycolysis, this whatever NADH we are going to form here, that is going to be reoxidized here by a lactate enzyme, that is called as lactate dehydrogenase. So there we are having a similar type of process, another dehydrogenase enzymes which works for the reconversion of NADH to NAD plus. So these are some of the common example of, you can call it dehydrogenase enzyme. Then we are having second category, that is called as transferases. Transferases are the class of enzymes that normally transfer a specific functional group from one molecule to the another. The molecule which comes from one molecules, that is called as donor molecules, and where it is going to be added, that is called as acceptor molecules. Like in this case, AB is going to react with C. So because B is going to be transferred from AB to C, so this molecule is called as the donor molecule, and this is called as the acceptor molecule, and B is ultimately transferred from AB to C. So A becomes separated alone, and C becomes BC. Transferases are implicated in hundreds of different biochemical processes, biochemical pathways, and are essential for some of the most common processes of our lives. Transferases participate in a number of cell reactions and also utilized during the translation reactions. Mechanistically, an enzyme catalyzing the following reactions would be considered as an transferases. The nomenclature, when we are talking about the nomenclature, how these transferase enzymes are going to be named. So systematic names of transferase are based on the donor, acceptor, and group transferase. For example, methylamine, L-glutamate, and methyl transferases. In this case, this is the donor molecule, and this is the acceptor molecule, and this is N-methylamine. Methyl is the group which is going to be transferred, and this is the suffix. So donor, acceptor, group, and then transferases. So in this case, transferase methyl glutamate and transferase, where methyl transferase is the electron category, methylamine is the donor, and glutamate is the acceptor. Nonetheless, the more frequently used nomenclature of transferase are often forms acceptor group transferase or donor group transferases. So these are the two another types of nomenclature we are having, which by how we are going to have named. So this is one way, this is the second way, and this is the third way how we put the names. These are the different subcategories of the transferases enzymes. So we are having a number of classes. If single carbon transferases, which contains functional group of hydroxy, methyl, carboxys, carbonyl, formyl, or amino transferases. Then LDL or ketone transferases, acyl transferases, transfer glycosyl, hydroxyl, and so different groups we are having depending on which type of molecule, which type of groups is going to be transferred from one place to other place. The application of this transferase enzyme are two common transferases are terminal transferases. Normally, this terminal transferase is one of the few DNA polymerase functioning without an RNA primers. Normally, almost all the DNA polymerase enzymes requires a small primer molecules, but this terminal transferase does not require any kind of primer. So they add transferase, that is one of the most very common enzymes used during the genetic recombination, or genetic, you can call it the RDT, recombinant DNA technology. Then second, another enzyme is glutathione transferase. With high diversity can be applied in the plant to segregate toxic metals from the rest of the cells. Then glutathione transferase could also increase the resistance in both biotic and abiotic stress reaction, and this can also explode as a target for anti-cancer medications. So these are some of the application of transferase, common example of transferase enzyme. The third category is hydrolase. Hydrolytic enzymes that are commonly used in biochemical catalysis, utilizing water to break chemical bonds in order to divide a large molecule into two smaller ones. Hydrolases are pivotal in the body since they digest large molecule into fragments. Some hydrolases could release energy also when they take effects. A number of hydrolases, especially proteases, are connected with the biological membranes as peripheral membranes, proteins, or affixed through the single transmembrane helix. Some other are multi-span membranes also. Nomenclature of these hydrolases are substrate hydrolases or substrates. This is the common reactions where a molecule is AB in presence of water. It is going to be hydroxylated. So this A becomes AH and B becomes BOH. So this is in the presence of water, AB has been hydrolyzed. These are the roughly different classes of sub-classification of hydrolases. So because this is the third class, so the first subclass is 3.1, which cleaves the ester bond. 3.2, which acts upon the sugar. 3.3, which cleaves ether bond. 4, destroy peptide bond. So depending on again different type of chemical reactions, we are having a different classes of hydrolases. The common examples are almost all the digestive enzymes are hydrolase enzymes. Other than that one, this is expressed by lactobacillus jenseni in the human gut could stimulate liver to secrete bile salts, which facilitate the digestion of foods. Acyl peptide hydrolase, that is another hydrolase found in the human erythrocyte, could potentially treated as a biomarker for low dose exposure to organophosphate. Glycoside hydroxylase play a unique role during the various biological processes such as metabolic of cell walls, biosynthesis of glycans, signaling, plant defense, and metabolic processes of reserve. Then we are having leukotrienes A4 hydroxylase, which could act upon the epoxide in the final step of the biosynthesis of leukotriene B4s. The leukotrienes, along with the prostaglandin, thromboxanes, these are the lipid mediators. This leukotrienes A4 hydrolase acts as the bifunctional zinc metalloenzymes, and that is the one of the important enzymes in the 5-lipoxygenase pathway. Nucleoside hydrolase plays a central role in the purine salvage pathway and also functions as a primary target for the exploration of antiparasitic drugs. Fourth class is lyases. In physiology, lyase is the enzyme which catalytically aiding the breaking various chemical bonds by means of the elimination reactions or other than hydrolysis or oxidation. So it removes certain groups without involvement of water. This reactions often results in the formation of new cyclic structure or a new double bonds, and it is a reverse reaction called Michael additions. To obtain either a double bond or a new ring, lyase acts upon a single substrate and a molecule is eliminated. Lyase can be commonly observed in the reactions citric acid cycles and in the glycolysis. These are named as substrate plus group plus lyase, such as decarboxylases, dehydratases, or aldolases. Uh, Aldolase, probably you may remember during the glycolysis, where fructose 1,6 bis phosphoglycerol is going to be divided into two fragments, 1,3 bis glycerol 3 phosphate and hydroxyacetone phosphates. That aldolase is lyase in them. These are the total 7 classes of lyases, and depending on their functions, we are having total 7 classes. So, first class is which cleaves carbon-carbon double bond, including decarboxylases, aldehyde lyases. Group 2, which contains groups of lyase that break carbon-carbon double bonds, such as dehydratases. Then group 3, which will release ammonia. Group 4, which releases H2S. So, similarly, on the basis of other functions, other molecules, which type of molecule they eliminate. So, we are having 7 classes of lyases. Then we are having isomerases. Isomerase present in the many variety can generally be divided into structural isomers or stereoisomers. Normally, if you remember the isomerization reactions, we are having two types of isomers: stereoisomers or structural isomers. Structural isomers have a different sequence or different connectivity of bonds from one another, whereas stereoisomers have the same order, same molecules, but in three-dimensional orientation, three-dimensional arrangements are different. Intramolecular lyase, oxidoreductase or transferase in the subcategories of isomerases, catalyzes the interconversion of structural isomers. So these intramolecular lyase, oxidoreductase or transferases, they normally leads to structural isomerization reactions, whereas racemase, epimerase, and cis trans isomers promotes the interconversion of stereoisomers. The prevalence of isomer in nature is partly dependent on the isomerization energy and the differences in the inner energy between the isomers. So these are the different classification of the enzymes. So isomerases have been assigned EC number of EC5 and are further classified into different category. One is racemase or epimerase. Isomerases in this class includes these two categories, both of which invert stereochemistry of the target chiral carbons. Racemase mainly acts upon molecule with only one chiral carbon of the stereoisomer conversion, whereas epimerase normally targets the molecules with multiple chiral carbon. Then we are having cis trans isomers. Cis trans isomerases normally this can distinguish by the absolute configuration rather than by the position of the substance substituents group with respect to the planar substance. Then we are having another category, that is intramolecular oxidoreductase, then intramolecular transferase, then intramolecular lyase. The main function of these isomerases are application of isomerase in sugar manufacturing is the most common, like glucose isomerase, which catalyzes the transformation of D-glucose to D-fructose, which is the key part in the high fructose corn syrup production and result in high yield of fructose with minimal side products. Then we are having another interconversion of sugars, where we are going to have use this isomerase enzyme. Then sixth class is we are having, that is called as ligases. Ligases is the ligation reactions. In the biochemistry, ligase is also called synthetase, and that is an enzyme catalyzing the joining of two more large molecules through the formation of a new chemical bond, such as carbon oxygen, carbon sulfur, carbon nitrogen, or the linking together two compounds, usually accompanied by the hydrolases of a small attached chemical group. This procedure usually assimilates the needed energy from a cleavage or the energy-rich phosphate bonds, involves the conversion of chemical energy, or offers a linkage between the energy requiring synthetic process or energy linked breakdown reactions. Because whenever you are going to have a bond formation, you require energy, and there should be a supplement of the energy production energy. So energy should come from some places. So this energy either comes from the energy rich phosphate bonds, or some kind of bond is going to be broken, and that bond breaking is going to release energy, and that energy will be used for new bond formations. In most of the cases, the simultaneous conversion of ATP to adenosine diphosphate ADP functions as the source of energy. Like in this case, if you will see, this is the molecule AB, and this is the XTP. X means adenosine triphosphate or guanosine triphosphate or cytosine triphosphate. So this AB will be joined together, but in the process of this one, it requires energy, a bond formation for this one, and that energy comes from the breakage of this phosphate bond. Total 6 classes we are having for the ligases, and these are the 6.1, 6.2, 6.3 to 6.6, depending on which type of new bond is going to form. The function of this ligases are one of the common example of ligases, DNA ligase, which is a common example during the joining of the two DNA fragments or two DNA molecules. Then we are having T4 RNA ligase 1, which is capable of catalyzing the ATP dependent covalent joining of single stranded 5' phosphoryl termini of RNA to single stranded 3' termini of RNA. Then we are having ubiquitin ligase, which commonly called as the ethyl ubiquitin ligase, that is enzyme existing in single polypeptide or a multipolymeric complex. This enzyme, this ligase, could work together with ubiquitin activating enzymes E1 and ubiquitin conjugating enzyme E2, which is a part of proteasomes. And the last class we are having, that is translocases. This translocase is the seventh newly added classes, that is a general terms for protein that assist the moving molecules usually across the cell membrane. These enzymes normally catalyze the movement of ions or molecule across membrane or their separation within the membrane. This reaction is designated as a transfer from side 1 to side 2, or because the designation in and out which has previously been used can be ambiguous. So the enzyme, if you see this seventh classes, the enzyme classification and nomenclature list was first approved by IUB in 1961. That time we had only 6 classes, E1 to E6. However, it was not noticed back into the none of these classes clearly describe this type of translocation enzymes, the enzymes which are involved in translocations. So that's why translocases enzymes has been newly added classes. Translocases are the most common secretion systems of gram-positive bacteria. Translocases have different classification classes. So they have their classification is on the basis of which type of molecule is going to be translocated. So their subgroup 7.1 is catalyzing the translocation of hydrones. 7.2 catalyzing the translocation of inorganic cations, then organic anions and chelates. So depending on which type of substance is going to be transferred. The further classification concerns the reaction that provides the driving force for this translocation, means from where the energy comes. So that is linked to oxidoreductase reactions, hydrolases of nucleotide triphosphate like ATP, GTP, CTP, TTP, and then energy may comes from the hydrolases of diphosphate, or energy may comes from the dicarboxylation reactions. So these are the source of energy. So every class will have a subcategorization on the basis of the energy source. The function of these translocases are normally, sorry, translocases play an important role in the biological activity because of their critical functions. Most of the protein needed for mitochondrial functions are encoded by the nucleus, because most of the proteins are genes. Almost all the genes we are having inside the nucleus. So most of the proteins are going to be coded by the nucleic acid, which is present inside the nucleus. Now, translocases of the outer membrane can work in conjugation with the translocases of the outer membrane, inner membrane, to transport protein into the mitochondria. In this case, we are going to have a pre-protein translocation into the mitochondrial matrix, requiring the pre-sequence translocation to the inner membranes and the pre-sequence translocation associated motor. Carnitine translocase is responsible for the translocating both carnitine fatty acid complexes. If you remember the fatty acid metabolic processes, so normally fatty acid synthesis takes place into the cytoplasm. So we are having fatty acids are inside the cytoplasm, but fatty acid metabolic process occurs inside the mitochondria. So fatty acid need to be transported from cytoplasm into the mitochondria for their metabolic processes, for their metabolism. Directly this fatty acid cannot be transferred. So there we are going to have this translocases functions which transfer the fatty acid, fatty acyl molecules, which with the help of this carnitine molecule. So for the fatty acid transportation we require this translocase enzymes. Then we are having ADP, ATP translocase. They normally transport ATP and ADP from across the membrane of the cytosol. Then we are having ADP transporters. Then we are having DNA translocases of the motor protein that convert the chemical energy of ATP hydrolysis into the divalent movement along the DNA. So these are the total 7 classes of enzymes, their sub-categorization and their main functions. Hope you have understood this detailed classification of enzymes roughly. Normally, normal UG student does not require this many subcategorizations. So if you remember the top 7 classes, their functions and their basic chemical reactions and some few examples of them, that is sufficient for you. So in our next video, we will come with the some factors which normally affect the enzyme activities or enzyme catalyzed chemical reactions. So thank you very much. Have a nice day. Thank you for watching. If you're not subscribed my channel, you can subscribe it. You can share with your friends. Have a nice day.